some of the characteristics of the different phyla of the invertebrates and comparing them. We're also going to be making some cross references to some of that big chart that I gave you. And I wanted to ask, I think there was a question before we left. What was your question, do you remember about, did you say something about the chart? No. Okay, the chart review so far. I've asked for me to be stopped about around 30 minutes from now, so that we can return exams and get you the answers and let you do the test corrections for Monday. I think that might have been a question before. Not necessarily, but I think that if you go from top to bottom and compare them from side to side, you'll see that it will be a very helpful chart. I'm not saying you have to have everything on the chart. What I would do as we go through the various groups would be to look at that chart and see which things I'm emphasizing. If you want to use a highlighter or whatever, in fact, that might be very helpful to do. I would also like to remind you that there is a quiz on Friday, and it would be over viruses, the overview of animal diversity, and chapter 33 invertebrates. So it could be quite a bit of stuff. You will not have anything about vertebrates. Specifics will not be on it because we'll start talking about it after the quiz. Okay, any questions about the schedule? And I'm not sure when our test is for the section, but it will be coming up eventually. Okay. Any other questions? Yesterday, now that you don't have enough to, for references and stuff, don't get excited. I added something yesterday to the exam study sheets, C33 invertebrate lecture visuals. That's what I'm going to be doing today. So it is there. So bring that up. What is the basal phylum, the lowest of animals? The sponges, and their phylum name is Porifera because they have all these pores. If you think of a real sponge. And really, okay. So what we have here, there's a picture of a sponge. There are freshwater and there are marine sponges. Now, again, I would take a look and have this. I know it's very fine and it's very small, but I would probably have this out. Phyloporifera pore-bearing, defining traits, and they've got color cells, which are called chromocytes. Basically, what you have is an outside epidermis with a hollow inside. You have one opening here, but you have all these pores where the water comes in from the sides, and as you see, it comes in from the side and then goes up and out. But through this little layer of cells, and we're not even going to call it a tissue. It is basal, lowest, but it has some specialized cells. It has amoebocytes, which are almost like amoebas that are running through these few layers of cells on the side. They help trap, catch any food. Basically, what they are filter feeders or suspension feeders. They don't. The other thing about sponges is they're completely sessile. once they become an adult, they stay attached to the same spot for the rest of their life. Doesn't that sound exciting? Okay, so that's probably something that we can work on improvement. There are sort of calcified type structures which actually provide the structure form. Those are called spicules. So spicules are part of the skeleton, if you want to call it that. Also inside of this layer of cells are these choanocytes. And the choanocyte is very similar to look at this. Whoa! A choanoflagellate, which is a protist, and we've already learned that the choanoflagellates are thought to be the ancestors. That's a big claim to fame. The ancestors to all animals. And here you actually have choanocytes on the inside there helping to absorb food particles. And phagocytosis, because of the amoebocytes, see the amoebocytes and the choanocytes, and it keeps the current of water. So they're sort of like filter feeders, comes into the side, and these specialized cells try to trap the food, and then digest it, absorb it there. Very simple, but it is a multicellular animal. And so they call it the mesophyll, which is sort of the cellular layer. There's a little epidermis on the outside, these cells on the outside. But again, we're saying no real true cellular level of organization, but not tissue. There are many different types of sponges. If we look down through here, the classes you do not need to know. Okay, so down to the bottom, you do not need to know the classes for the different types of skeletons. They can reproduce asexually by budding, regeneration. They can also reproduce sexually. Most of the sponges are hermaphrodites, which means they have both male organs and female organs. And they release their eggs as sperm into the water, and there's fertilization. And then once it attaches, it grows and it's sessile. So sessile means doesn't move. I would catch that term. I'm not sure if it's on this chart or not, but sessile is a condition of remaining attached in one spot. So they don't really truly have a digestive system. It's just an area where food is trapped. Fusion. So no civilization, which we can tissue or anything. Developmental mode comes from two germ layers, diploplastic. And those are some of the, so spicules would be something I would say makes up the skeleton. And I've introduced the word sessile for remaining attached and not moving. Any questions on Porifera? Okay, let's go to the next grouping that's called Cnidaria. There's a C in front of it. Cnidaria. Their main thing is that they have these stinging cells on the ends of the tentacles. They have a radial symmetry, and oh, by the way, I forgot to tell you, sponges have no symmetry. They're really considered to be asymmetrical. They can grow in all kinds of weird shapes. Okay, whereas we have radial symmetry with the Cnidarians. What are we talking about? We're talking about jellyfish, we're talking about hydras, different hydrozoans, sea anemones. These are all Cnidarias. So there are two main body plans. Let's just bring it down here. Two body forms or body plans within this group. The The more stationary type looks more like a sponge, but it's not completely stationary. It's called a polyp form. It's a life form. And then the other is if you kind of turn it upside down and you have the bell shape with it, like the jellyfish, with the tentacles down in the water, the jellyfish. So it's Medusa. So you have polyp of Medusa being the body forms of those cnidarians. But they have stinging cells called nematocysts on the end of the cnidocytes. They do have a tissue level, so they're the first group going up to have tissues, but they're missing cephalization. These are cephalizations like brain, the formation of like a brain. They have what we call, some of the complexities is that they have like a nerve net coordination. So there has to be some spatialization and communication, because what happens if you're standing here and you're a hydra, which are found in freshwater streams, jelly-like, if something touches a tentacle, it actually communicates with the rest of the tentacles, and they all come over to help pull the food into the gastrovascular cavity. So gastrovascular cavity is a digestive type cavity. One of the drawbacks of this group is we continue to go with only one opening. The mouth is also the anus. That may not sound real delightful. Okay, but nerve net coordination with stinging nematocysts, polyp medusa forms, gastrovascular cavity, which is where food is, digestive tentacles, mouth and anus single opening. So this is kind of a condensed version of some of the main points. I don't know that you need to necessarily have to be able to reproduce this, but it just shows you it goes through a polyp. Some go through both polyp and medusa stages, but some stay in the polyp stages, and some will stay only in medusas. So it actually shows some of the sexual reproduction parts. Okay, cnidarians, that was it. Platyhelminthes, the flatworms. They're actually next in terms of advancements. You actually have this, have bilateral symmetry. You have a right side, you have a left side. This is a free-living planarian. It's one of the examples that's given. In fact, if you go down to the classes, by the way, classes for cnidarians, you don't have to know. I would know the classes, the first three classes of Platyhelminthes. Turbellaria is the free-living worms, which would include the planaria. Trematodes are the parasitic flukes, and Cestodes are the tapeworms. Don't worry about the other fish parasites. So, they're bilateral. This is also the first group to be triploblastic in terms of development, which means they come from ectoderm, mesoderm, and endoderm. The cnidarians were diplo. They're still protostome. Well, they are protostomes. So really, in terms of, you don't talk about protostomes and deuterostomes until you get the three-cell life. They're acoelomate, which means they have no coelom, true body cavity. They're flat, like they're smashed. They do have, as you see with the free living, you see that there's actually a little bit of ganglia in the anterior part. And that's a ganglia is where you have a bunch of nerve cells hanging out, which can be the start of cephalization of a brain. Do you all know the positions for bilateral animals? Like if you're an animal, pores are crawling like this. The end of the animal, which meets the environment first, is the anterior. The end of the animal, which meets the environment last, is posterior. The surface of the animal, which normally faces downward towards the ground, is the ventral side, and the surface of the animal, which normally goes upward, is called the dorsal surface. And I communicate a lot in the anterior end, the dorsal side, or the ventral side, or the... So please be sure to know which those refer to. They also have a gastrovascular cavity. And look, they have nerve cords. You know, like our spinal cord, but it's not that complex. They actually have cords for nerves. Okay, catch this claim to fame statement. If there are nerve cords present in invertebrates, they're located where in the body? Which surface? Dorsal. At least the lower ones. Okay, I didn't word that right. The lower invertebrates, when you're in the nerve cord, it will be ventral. And ours is dorsal. So kind of watch for that. It's a ventral, it's like a ladder-like nerve cord situation. Ventral to this cute little planarian, he's got like an arrow head. There are eye spots there too. He or she has the mouth coming out. There's like a little pharynx tube, which like suctions up stuff from the bottom of the stream in which the planarian lives. So this one's in the coronal area. Flukes. Flukes are in the class Trematoda, and a fluke is an internal parasite, not free living. And the main claim to fame about this is that it requires an intermediate host for it to be transferred like to people, which could be the ultimate human host, but it has to spend part of its life cycle in a snail. So if you want to eradicate flukes, one of the things one would do in an area where fluke diseases are really prevalent would be to try to destroy, doesn't sound good from an environmental standpoint, they try to destroy and eliminate snails, because you have motile larvae that come out from the snail once it's spent some time in there, and then when the person's in the water and doing stuff, then the flukes can actually get into the human host. The third class, one of my favorites, just so gross looking. The class Cestoda, CES-TODA, and this is a capeworm. And there was a time, even that I remember being on a farm, that we still worried about tapeworm eggs and kids getting tapeworms, things of that sort. Some things to know about the tapeworm is, of course, it's a parasite. It's an internal parasite that lives in your intestinal tract, and it lays its eggs in your feces, and then when you defecate, then the eggs are passed on. Then if people don't wash their hands, or if they're preparing food, then it can be transferred to somebody else, so the eggs be ingested. Okay, now these long ribbon-like, and that's what they are, ribbon-like, and they can be 10 meters long sometimes. They can be really long. They have this really gorgeous head called a scolex. Looks like something from outer space maybe, but it's got hooks, and it's got suckers, and a way to hang on to your intestines while the feces go through. And it hangs on, but it can still produce eggs in these repeating sections of reproductive organs called proglottids. So proglottids are the repeating units of the tapeworm, through which the eggs can then be released. So that is also a platyhelminthes. As you can see, it's very fine. Okay, any questions on platyhelminthes? So triploblastic. There are free living. If I asked you what the free living example of a unknown that these words, you would say it's a planaria. If I ask you, for example, of a trematode, you would say it's a fluke. Its intermediate life cycle is a snail. The tapeworms could be very long. They have proglottids, scolates, suckers, and they're in cystota. Okay, I just threw these in here, but we're not going to really talk about them. I want you to know what rotifers are. There's a phylum Rotifera, a rotifer in the water. They are a multicellular animal, and sometimes when you get pond samples, we find rotifers. They're very exciting. I get really excited when I find a rotifer because it's just kind of like a motorboat type thing, but it's got this crown cilia. Up here, it actually has jaws all the way back here. And you can see it does have a stomach and so forth. But this is a rotifer in the water. It has a crown of cilia with jaws. And this is how big it is. This is 1/10 of a millimeter from here to here. So it can be close probably to a millimeter. A little shorter than that. Okay, phylum. Okay, remember this is out of order. I went ahead and did it this way. Now that we know that they're in that existence grouping we talked about on Monday, but I'm going to talk about nematodes here anyway, even though they're really supposed to be talked about later. They're unsegmented roundworms, so that's a round worm. It is round. It doesn't have segments around it. And they're mostly parasitic except for like free-living vinegar eels that you can find in the soil and other places. And the hookworms, which we know that our dogs and cats and other animals can get. Ascaris is one of the famous roundworms that's also a parasite. Trichinella is that trichinosis from pork. What you really have, you do have subtilization. They're still protostomes. They actually, whereas flatworms had acoelomate, these have pseudocoelomate. So I would probably do something and mark that. So they're kind of in between. It looks like they have a coelom, but they don't. Remember with the other first chapter, we showed you the difference between acoelomate with no coelom body cavity and a pseudocoelomate. Let's see. They have hydrostatic skeletons, which is that water pressure helps to keep their form. Okay, a big deal. They have an anus. You've actually got a mouth, and you've actually got intestines, and then they have an anus. So that's kind of a big deal. One of the first groups to have that. I mean, think of your options. Okay, there's still diffusion. You don't need to know the classes. Non-living cuticle for outer covering, which is why they're in that thesis group for molting. They're important to decomposing, and they're predators of microbes, so they will actually eat microbes too. So. Nematoda. Phylum Mollusca. Think of soft body vertebrates. More complexity. We're going up in complexity. The three main parts of a mollusk, which you may want to add, and what's up here on the defining traits: head, foot, and a visceral hump or mass. Visceral hump or mass is where a lot of the viscera, the organs are. So a head, a foot, a visceral mass. And the foot is a really specialized structure, often having to do with movement, but also aiding in the capture of the food in some way. There are three different classes of mollusks that we're concerned with. This is Gastropoda. snails, gastro means stomach. It looks like its stomach footed, crawls on its stomach. So we're talking about snails and slugs. One of the other specialized structures of this group is most of them have a radula, particularly the snails. The radula is a scraping tongue. If you've ever had an aquarium and you have snails going up the walls of your glass aquarium and you watch that scraping, the scraping tongue, very sharp, that scrapes the algae off. So it has a mouth, esophagus, and now we're starting to have real digestive organs, digestive tract. You actually have an intestinal loop. The anus comes out here in the back. You have, this is from here to here is the whole visceral hump or visceral mass. The mantle is the membrane-like structure just on the inside of the shell. Gastropods have a shell, unless they are a slug. All right. Snails. The mantle is a specialized mollusk structure that secretes the shell. And they have gills back here. And there's a slight nerve cord, some ganglia up here. Okay, then, one we're probably even more familiar with are clams, bivalves. Valve is a shell, bivalvia, so these would include oysters, scallops, clams, mussels. And there's a hinge area here. There's a huge muscle here and a huge muscle here. These are adductor muscles that keep the clam up. Okay, that they close, and it's hard. You have to cut these muscles. If you're eating them, you have to cut those muscles to release, because they shut. They really are shut. Okay, so you've got a mantle cavity where you have the gills. You've got the water flow coming in from the posterior part. It goes in. By the way, it's a characteristic of mollusks, and particularly octopus. You see where the gonad's a reproductive organ. It's actually, we're seeing a heart here. It's a dorsal heart. A coelom, body cavity. So, they are the first, mollusks are the first coelomates. And you can look at some of the rest of the structures. And then cephalopoda, head-footed. In other words, the foot coming off the head. They have eyes. They have simple, not as in, not advanced, but simple one eye versus compound eyes of an insect. They also, the octopus has been involved in a lot of learning experiments, that you can put something that it normally eats in a glass jar and have it closed. Okay, that's about the time. Okay, give me a couple more minutes here at least. And you can watch videos of this. The octopus would actually use learning, problem solving to try to open. So probably so far these are the most intelligent of all of the organisms we've talked about so far. And the chambered nautilus, the squid, the octopus, and the nautilus. The earthworm is in the phylum Annelida, segmented earthworms. You will want to eventually know for lab in particular all these different parts. They have a digestive system. They're really regarded to be pretty complex. They have organ system, ventral nerve cord, another hydrostatic skeleton, circular longitudinal muscles, a complete digestive system, diffusion through the cuticle, some different analytics. system. Okay, so far, up until now, we've not had a closed circulatory system, which means blood is confined within a series of blood vessels. A lot of these other organisms, it's just the fluid just flows over the inside of the cavity. Okay, and I am going to try to wind this up quickly here. Here's a leech, which is also a member of the Annelidas, segmented. You can see the circles. Then we get to the phylum Arthropoda. We've got the crayfish crustacean, which has two body regions, a fused cephalothorax and an abdomen back here. It's also in a class called pancrustacean, where they're adding insects and crustaceans together now. They have jointed legs. Arthropoda means jointed legs. Spiders, class Arachnida, or sometimes called Chelochera. Two body regions, cephalothorax and an abdomen. Four pair of legs. You've got the spider family, the scorpions, horseshoe crabs. You've got true spiders. They have poison glands up by their chelocheras, which are like their front little appendages that can bite you. Injects poison to do that for their prey in particular. They're very studious lungs. They have spinnerets back here to spin the web. The webbing material is liquid as it comes out, and then when it hits the air, it actually turns to a solid. You have seen very elaborate webs. And then the grasshopper is an example of an insect with three body regions, three pair of legs. We're not going to forget the example. Myriapoda, centipedes, millipedes. Remember, one pair of legs per segment in the centipede, two pair of legs per segment in the millipede. Which one? Oh, by the way, the centipede is carnivorous. The millipede is herbivorous, which means it lives on plant material. So which one do you think bite can most hurt you?
Hunting.
Carnivorous one. I said which one most likely to bite you would be millipedes. Are more dangerous to us. Metamorphosis. This is complete metamorphosis. An example of incomplete is the grasshopper where you have eggs hatching into nymphs. Nymphs are immature forms. Many moltings in the adult. The stressing of how insects are so important for pollinators and pollination of flowers. Deuterostomes, so then we get to the deuterostomes, so then guess what the last group is? Radiocleavage, formation of the anus from the elasticor. We get to the last phylum, which is the phylum Echinodermata. And here we have the starfish, spiny-skinned invertebrates. They have a water vascular system, which is very unique. It's the only group that has a water vascular system. It's like an indoor plumbing, piping system, taking water in and out, and using suction because there are rows of suction cups in the 2 feet that helps the clams. They squirt the water out. They bring water in from the top of sieve tube, sieve plate, and then releases it. But they're strong enough to open up clams. And then what happens is how they feed. That's really neat. If you watch how a starfish feeds, its whole, see this whole circular central disc area? That's where the stomach is. The stomach actually descends down into the body of the clam, secretes digestive enzymes, dissolves the viscera of the clam, and soaks it up. Right, very much like the fungi. Yeah, it's a really neat type of deception. Okay, I'm going to stop there, and we will do 16 false, 17 B as in best, 18 D as in dog, 19 endospores, ENDO, endospores, 20 disinfection, 21 C, 22 true, 23 A, 24 false, 25 B as in best, 26 C, 27 histology, 28 mycology, NYC, 29 silica glass, something like that. So add up the number I marked wrong with a line in that column and make sure that it matches what I wrote at the bottom of the column. Any problems or questions? See me.
Oh, whoopsies.
Okay, second column.
That was silly.
E. coli, E, and then COLI, or the Escherichia coli. 31 false, 32 B as in best, 33 B as in dog, 34 autoclave, 35 eukaryotic, 36 B as in best, 37 C, 38 sporophyte, 39A, 40 B as in best, 41 B as in dog, 42 C, 43 flagella, 44 pseudopodia, 45 cilia, 46 B as in dog, 47, the best answer was food supply for the embryo, 48 megaspore, 49 true, 50 and 51 are both false, 52 circle or ring, 53 scattered, 54 true, 55 vascular cambium, 56 D as in dog, 57 is ground, 58 is B as in best, 59 is A, and 60 is dicot. Okay, add up the number wrong in that column. Then you have the opportunity to get 3 extra credit questions that would cancel out 3 wrong. So do the math, add up the number wrong for column A with the number wrong for column B. I put it at the top, minus whatever. Then I added plus however many you got for your extra credit. Came out with a total negative that you missed. Each question, since there are 60 questions and it's worth 150 points, each question was worth 2.5 points each. So however many the net list is times 2.5, that number then subtracted from 150 is how you got your score. Now, the original day of testing, these are the scores that we had. Pretty impressive. Because of the extra questions, we have people above 150. Congrats, and all these A's, that was pretty fabulous. And then the B's, 127 through 120. And so that's how the original testing went. There may be some additional makeup scores that aren't on there right now, but it gives you a good idea of where you fell. So congratulations.